Good day to all of you. Through this video, we'll discuss the cellular basis of the spermatogenesis, which is a part of the gamete biology in SASI 11 Unit 1 in semester 3rd syllabus. So the male gamete is known as the spermatozoa or the sperm. A sperm or the spermatozoa is an elongated microscopic structure differentiated into the following main parts, that is head, middle piece and the tail. The process of the maturation of reproductive cells of testes in male and formation of the male gamete or sperm is known as the spermatogenesis. The entire process of spermatogenesis has been separated into the following step, that is formation of the spermatid from the cell of the germinal epithelium and metamorphosis of spermatids into sperm or spermiogenesis. So it is a figure of LS of human sperm in which the three part is divided. The first one is the head part, then the middle part and the tail part. End with the end piece. So how the formation of the spermatids occurs. So the entire process of the formation of the spermatids from the primary germ cells is completed in three steps. First is the multiplication phase, then growth phase and then maturation phase. So multiplication phase, the primary germ cells multiply by mitotic division and produce the spermatogonia, and each spermatogonia possesses diploid number of chromosomes. The growth phase is a product of the multiplication phase, and in this the increase in size by the accumulation of the nourishing material, and finally it attains the maturity, and the mature spermatogonia are called as the primary spermatocytes, which is also diploid in number. Then the maturation phase. During this stage, each primary spermatocyte divides by reduction division, which is called as the first maturation division, and the product two haploid daughters called as the secondary spermatocytes are formed. The secondary spermatocytes again undergo the second maturation division, which is each spermatids metamorphosize into sperms having a long tail and head with nucleus. Then the metamorphosis of the spermatid is known as the spermiogenesis or the spermatiogenesis. During this process, there is a gradual differentiation of the non-motile, that is stationary, rounded spermatid into an active and motile spermatozoa or the sperm. The spermatid forms as a result of the maturation division is a typical animal cells having cell organelles like mitochondria, Golgi body, centrioles, and haploid nucleus. During spermiogenesis, there is several changes like changes in nucleus size, nucleus shape, centriole, changes in the mitochondria, formation of the acrosome, and the loss of cytoplasm. So RNA and other accessory non-chromatin materials are removed, and only the hereditary material, that is deoxyribonucleoprotein, that is DNA, are left in the nucleus. One of them enters into the depressed portion developed in the posterior part of the nucleus known as the proximal centriole, and the other centriole called as the distant centriole, which lies behind the proximal centriole. So changes in the mitochondria, so axial filament between the distal centriole and the ring centriole in the middle piece of the spermatozoon. So there is a spiral sheet provide energy and strength to the sperm when it swims or moves towards the ovum. Formation of acrosome is formed from the Golgi complex of the sperm, which forms the cap around the nucleus. The maximum amount of the cytoplasm of the spermatid is lost, and the remaining part of the forms a condensed layer around the periphery of the spermatozoon, which is called as the manchete. So here is a short description of the spermatogenesis, and each day about 300 million sperms complete the process of the spermatogenesis. So here is a flow chart of the spermatogenesis. The primordial germ cells enter into gonads known as the spermatogonium. Then the mitosis, there is a diploid number of the chromosome, and then the primary spermatocyte or the mature spermatogonia are formed, and then the first meiotic division occurs, form the secondary spermatocytes. Then the mitotic division forms the spermatids and differentiation to form a mature sperm. So process of spermatogenesis and spermiogenesis. The figure of the seminiferous tubule showing the spermatogenesis. Leydig cells provide the nourishment to the developing sperm, and there are different stages of the sperm, that is spermatogonia, spermatocytes, spermatids, and the sperm. So this is all about the notes on the cellular spermatogenesis. So thank you and keep watching.